everyone. I'm Shreyasi, and today I'll be addressing the question of whether or not dopamine is a stimulus if it is also a reward. Dopamine is one of the many neurotransmitters in the body, and it's associated with love, lust, desire, and addiction. There seem to be a lot of inconsistencies with classifying dopamine in the reward system, and various classifications of the dopamine has led to contradictory interpretations of experimental results. So before I dive in, the first thing I want to do is distinguish between a stimulus and a reward. So a reward is defined as that prize or that gift or that end goal that one receives after completing a designated task, whereas a stimulus, or in this case, a positive stimulus, would be defined as that expectation for that reward. So, looking at previous studies, one study done on rhesus monkeys showed that when the dopamine receptor antagonist glupentexol was provided to these monkeys, there seemed to be a decrease in the cocaine intake. Another study done on fruit flies showed that when the fruit flies were exposed to less food, food in this case would be defined as the reward, they seemed to extend their proboscis more frequently trying to receive that food. And then on another study on crayfish showed that when cocaine and amphetamine was injected into their systems, the level of movement had decreased. So cherry shrimp was used in this experiment because they are easily reproducible in the lab and they have no blood-brain barrier. So in this experiment, for each trial, we had four tanks set up, and inside each tank, we set up two dividers with three holes inside each of these dividers. In the experiment, we poured 10 liters of distilled water into each tank, and we also placed 2 milliliters of the appropriate liquid or solution into the designated tank, depending on which trial we conducted. So if I were to test haloperidol or dopamine, I would create that solution and place 2 milliliters of that solution into the experimental tank. So we had two tanks which were used for the control group and two tanks which were used for the experimental group. We also recorded the temperature of the room as well as the temperature of the water before conducting the experiment. Now, as you see here, we placed two small jars next to each tank and we filled those up with distilled water as well. And we collected 5 cherry shrimp and we placed them into these jars and we let them assimilate to their environment for about 15 minutes. After those 15 minutes were up, we took the jars and we gently placed the shrimp into each tank so that there were 10 shrimp in each tank. And so for each trial, we observed about 40 shrimp. And so what counts as a movement? That's probably one of the questions you would want to be asking right now. Well, if the shrimp moves through the hole once, we counted that as one movement. If the shrimp moved through the hole and back out again, we counted that as two movements. As long as the shrimp pokes its head through the hole, we counted that as one movement. So for our results, we first created a graph comparing dopamine with the control group. And as you could see here, dopamine seemed to have some sort of inhibitory effect. You can see with the control group, the shrimp moved about 5 movements per minute, whereas with dopamine, they moved about 2.5 movements per minute. When comparing the different treatment groups, we see that with the glutamate by itself, it led to about 10 movements per minute, and the shrimp exhibited stimulatory behavior. Now, when dopamine was paired with that glutamate, we see a decrease in behavior. Behavior was inhibited. Haloperidol didn't seem to do anything on its own. However, when it was paired with the dopamine and the glutamate, it restored some of that activity. However, the activity levels weren't increasingly high compared to the treatment group with just glutamate on its own, and that could be due to the fact that dopamine may have not been fully blocked at that time. And this graph also shows that dopamine exhibit allowed for inhibitory behavior. Glutamate allowed for stimulatory behavior, as you can see by this slope. And even though the slope between the control and dopamine were relatively similar, the slope for dopamine was still lower, suggesting that it was inhibitory. So dopamine is the reward in which the organism seeks in the reward system. And while observing the movements of the shrimp for a set period of time, it was determined that these results actually appear to contradict the standard dopamine model. So, as previously mentioned, a reward can't be defined as the stimulus, and understanding that there's a chance for that achievable reward is what initiates that effort to get there. So, activity can't occur if there is no reward, if there's an elimination of that reward. So, two examples I like to use is the office setting example and the donkey and carrot scenario. So, in the office setting example, if an employer is promised a paycheck, then they will want to work harder for it. The paycheck in this case would be defined as the reward and them working hard to achieve it and their expectation that they will receive that paycheck, that would be defined as the positive stimulus. For the donkey and carrot scenario, if the donkey sees the carrot just hanging in front of its face, then the carrot would be defined as the reward, and the donkey moves towards it knowing that it can grab the carrot if it just moves closer to it. That would be considered as the stimulus. So instead of identifying dopamine as an incentive, this new model examines that activity level of crustaceans and uses that to kind of define what dopamine is and kind of analyze other chemicals such as glutamate and haloperidol. So, as I mentioned previously, glutamates seem to encourage stimulatory behavior, and there could have been a stimulation in the olfactory senses. And since stimulants seem to correlate with the olfactory lobe, it could be reasoned that maybe the smell given by the amino acid glutamate is what triggered that hint or that possibility that there's some reward out there, and that might have led to an increase in activity. And when it was paired with dopamine, we saw that that level of movement was lowered, it decreased. So, referring back to the rhesus monkeys experiment that I discussed, high doses of the dopamine receptor antagonist seem to decrease the cocaine administration and that desire for food because the dopamine receptors were blocked. And because of that, dopamine was prevented from binding to its receptors. And the monkeys lost their desire or motivation to kind of go out and obtain that cocaine. Similar to the office setting example, I also like to talk about students studying for a grade. So if a student is promised that they can receive that A or that good grade, that A would be defined as the reward, and them studying for it and knowing that they can achieve that, that expectation that they can work hard and achieve that grade, that would be considered as the stimulus. So then what's the stimulus in this model? It could be reasoned that perhaps since glutamates seem to show that there was 10 movements per minute, there was a increased amount of movements, it showed stimulatory behavior, it could be reasoned that perhaps in this model, glutamate could be justified as the stimulant. Further areas of research could include the ADHD model as well as drug addiction. These are my references.